Ladies and gentlemen, may I all welcome you here at this inaugural lecture of Professor Douglas Hanahan, appointed as Extraordinary Professor on the TEFAF Oncology Chair in the Faculty of Health, Medicine and Life Sciences. The title of the lecture of Professor Hanahan will be The Daunting Complexity of Cancer. Understanding the battlefield is a step towards winning the war. Professor Hanahan.
Well, good afternoon. It's really a great honor to be here. I hope this microphone is on. All right. Yeah, so what I want to do is talk about an organizing principle to think about cancer. Because over 40 years ago in the United States, a war on cancer was announced, and with the thought that by marshaling our forces we could we could win the war. And 40 years later, it's clear we haven't won the war, although we're winning some battles. And so the question is, why is that? And so I want to give you sort of a perspective on where we are and how we might think about cancer, and if we could understand cancer better, maybe we could think about better ways to treat it. So what is cancer? Well, I think maybe I'll try to stick. So cancer is fundamentally a disease of uncontrolled cell proliferation. So, that in development, there's an orchestrated development. We start as a single cell, become hundreds of millions of cells that are working together in synchrony. But in cancer, previously well-behaved cells escape and begin to proliferate without abandon. And so this is, and what we see is the progressive development of lesions that the epidemiology and histology of human cancer suggest is a complex process. And so, for example, this is a schematic of breast cancer where the normal cells of the duct, that cells begin to proliferate inappropriately, producing what's called a hyperplasia, where there's aberrantly proliferating cells in the duct. These can become more aberrant, producing so-called dysplasias or carcinomas in situ that can then switch on angiogenesis and become more aggressive and eventually progress into invasive cancer that then begins to invade into the underlying stroma and then in due course produces metastases, which is the condition that is often proves lethal. So it's a disease of proliferation, but there's more than just expansion of the cells. So what we know is that cancer arises through, it's a disease of our genes, and that virtually every cancer cell contains mutations or alterations in genes that are in some way driving or enabling or allowing that cancer to progress. The what? It's not working? What? I was a bit worried about that. It should be out here, maybe. Yeah. Okay, is that better? Can you hear me? Okay. Okay, so basically, cancer cells or tumor cells have altered programs that cause them to stop performing their duties as sort of normal members of the society of cells that exist in our organs and tissues. And they become instead outlaw cells that are reprogrammed for proliferative expansion. And so what do we know about cancer? Well, the reason we haven't cured it is that cancer is not one disease, it's hundreds. It arises in hundreds of manifestations in virtually every organ of the body, multiple cell types within those organs. There are multiple subtypes, even in particular organ-specific cancers. There's hundreds of different genetic aberrations that can drive tumor growth.
and there's dizzying genomic disarray and histological variety. And so this is, the more we study cancer, the more we're faced with this reality. And so what is cancer? It's remarkably diverse, and the severity and pathologic consequences on an individual can vary dramatically, from relatively asymptomatic to rapidly lethal. There's unfortunately disconcertingly transitory therapeutic benefits for many forms of human cancer. And so that we've made, as I mentioned, we've won a few battles, but we certainly have not won the war 40 years into it. And so here's an example of melanoma, which is an often deadly form of skin cancer. And so here's a small melanoma. And this is really a poster child. This disease is a poster child for the opportunity and the promise, but also the challenge that we face in cancer. And that's basically because melanoma is a disease that kills you by metastasis. So the cells start off as melanocytes in your skin, and a primary tumor, like I just showed you, migrates to many places in the body. And so this is shown here, for example. This is an imaging of a patient that has metastatic melanoma. And so we've learned a lot about the nature of this disease. In fact, we heard a talk from Professor Peoples today about melanoma and some of the progress that's being made. And we know, for example, some of the mutations that are driving the disease. And this has led to the development of new therapies that target this disease in a very specific way, so-called magic bullets or smart bombs. So there's a new drug, a targeted smart therapy that basically melts these tumors away. And so these are actually normal imaging of the organs, but essentially most of this metastatic disease is melted away. You can see this here as well. The disease has largely disappeared. And so this is, in terms of fighting a war on cancer, this is what we dream about, is drugs that would melt away metastatic disease. And this is what we hope to accomplish. And so this is really, is a very, very exciting result. And it says that yes, we can identify causes of cancer, and we can target cancers and melt away disease. But this is also the illustration of the challenge we face, that cancer is a disease of aberrant cells that are essentially adaptable, mutable, changeable. They've lost a lot of the checks and balances that normal cells have to maintain their integrity of their genome and the integrity of their behaviors in single cells. And this is really realized here. So in 2 weeks, this metastatic disease largely melts away, but 6 months later, it's back. So this is the promise, and this is the problem, that there is rapid evolutionary adaptive changes that happen that render this therapy inefficacious. And so this really, I think, beautifully illustrates where we are, that we're understanding mechanisms of cancer. We can develop new drugs, but the cancers are rebounding in various forms of adaptive resistance. It's like trying to fight the Viet Cong. They're guerrillas. They adapt, they change, and that's what we're faced with. So basically, cancers develop and become increasingly aggressive in a form of Darwinian evolution, which selects for those cells that are the fittest at this illegal expansion. And so that is really what we're facing. And again, for the lay audience, I mean, most cancers consist of these assemblages of cancer cells that we call tumors. And so that was just one illustration. And the drug that works in melanoma doesn't work in many other cancers, even cancers that may have the same mutation of the same gene that was being targeted. So there's really extraordinary complexity at all levels: genetic, histological, pathological, prognostic, and therapeutic. And so the question is, so this is where it is rather depressing when you think about it, because we know so much and yet we know so little. And so the question can be posed: is there any way to rationalize this complexity? And are there common principles underlying this diversity? And so Bob Weinberg and I developed a hypothesis for rationalizing this complexity of cancer. And then the proposition was that disparate cancers share fundamental qualities, and that this daunting complexity really represents just different solutions to the same problem. That if you're going to become a symptomatic tumor, an outlaw cell that grows and progresses, that you have to solve a series of problems because the organism doesn't want you to do what you're doing. So cancer cells there are multiple barriers and roadblocks, we believe, that are used by the organism to prevent this aberrant behavior. And so successful cancer cells surmount multiple barriers and roadblocks used by the organism to prevent expansive cell proliferation. And so it's like trying to climb a mountain, and you can go up one way or another, and there's different solutions to how you might climb it. And different cancers do it in different ways, but arguably there are similarities to what they need to do to get to a symptomatic disease. And so this hypothesis is represented in this schematic here, which is a little discolored, but it's still clear, I think, and that is the notion that tumors acquire a set of common capabilities that are necessary to manifest themselves as a tumor. And so we posed this in 2000 in a perspective that was published in the journal Cell, and the idea was that tumors acquire these capabilities. And so I'm going to just walk you through what these capabilities are, and just in a simple manner explain why we think, what they are and why we think they might be important. Now, we went on a couple years ago and refined this notion, and there are now, we believe, 8 acquired capabilities that are quasi-distinctive and independent, and so I'm going to walk you through those. So, what are the hallmarks of cancer? Well, they're capabilities, functional capabilities that allow tumors to do something active, to do things that normal cells should not, and typically to do them chronically, rather than during the carefully orchestrated processes that happen during normal development, during normal homeostatic situations in the adult. Because again, cancer cells are not inventors. They're not inventing new things. They're simply co-opting natural systems that are used in other orderly processes in development and homeostasis. So the first hallmark is perhaps the most obvious for this. This is a disease of proliferative expansion of aberrantly expanding masses of cells, and so sustaining the signals for proliferative signaling is an evident hallmark. So the analogy is it's like the accelerator on your car. I mean, it's driving cells to go through the cell cycle and divide and proliferate and expand. There are signals that instruct cells to grow and divide chronically. So that is, I think, the first hallmark. The second one is really that there's a series of cellular breaks, of barriers to proliferation that are meant to render proliferative expansion transitory in normal situations. And these are called growth suppressors, and they're both intrinsic growth suppressors and community-wide growth suppressors. But the colors are not so good here. But anyway, so the notion here is, and what we now realize is that these growth suppressors can be removed, and so you basically lose the brakes that normally restrict cell proliferation. So the signals to stop proliferating are disabled. So you've got the accelerator on and the brake is off. Now, the third hallmark is another manifestation of barriers to aberrant behavior, and that is that we now appreciate that cells have an intrinsic ability to undergo various forms of assisted suicide or cell death, and of which the most prominent is a process called apoptosis. But there are varying forms of programmed cell death that normal cells use in various circumstances where it's deemed that they are aberrant and should be eliminated for the benefit of the larger society of cells. And so what we now appreciate is that many cancer cells develop to varying degrees ways to avoid this assisted suicide of abrogating this inborn willingness of cells to die for the benefit of the organism. And so this is another distinctive barrier avoiding programmed cell death. The fourth is another protective mechanism which involves the linear chromosomes and the realization that every time, by the nature of the double helix and the mechanisms that have evolved to replicate the chromosomes, that upon every chromosomal division, the chromosomes shorten at their ends briefly. It's called the end replication problem. And there are special structures at the ends of these chromosomes called telomeres. I won a Nobel Prize a few years ago. And what we appreciate now is that the telomere shortening is a counting mechanism that defines how many times a cell should be able to divide. And when the telomeres get too short, the cells are prevented from dividing. And cancer cells figure out a way to circumvent this counting mechanism, largely by inducing an enzyme called telomerase, which actually maintains the ends of the chromosomes in stem cells. Because again, stem cells of our body have a self-renewing capability, and they have evolved a mechanism to avoid this counting mechanism, and cancer cells co-opt that so as to avoid this cell division limit. So again, these are distinctive ways in which cancer cells are the three different kinds of barriers to chronic proliferation that cancer cells use. The fifth hallmark, it takes us outside of the cancer cell into the realization that as a nest of cells begins to grow and divide, it requires access to oxygen and glucose and other nutrients, and also ways to evacuate waste. And it's now appreciated that during the development, most solid tumors turn on angiogenesis, or the growth of new blood vessels. And that this is necessary, at least during early stage development, to feed and nurture the growing mass of cancer cells. And here's just, for example, this is a tumor here, and this is an aberrant vasculature which has been induced to supply this tumor. This is, for example, bits of normal vasculature up here. So this is just a graphic representation of tumor angiogenesis. The sixth hallmark is perhaps the least intuitive. The ones I just described, they're sort of intuitive of why cancer cells would want to adapt them or acquire them. The sixth hallmark, but is really undeniable, is the notion of activating the capability for invading local tissue and metastasizing to different organs. And again, tissue invasion and cell migration around the body are part of normal development. But again, they're transitory, they're turned on and off in very regulated manners. But in cancers, they can get turned on and persist. And so the notion here is that you end up with gorilla cells that grow by migrating and invading into normal organs, locally and throughout the body. And again, this may be a way, again, to live off fertile land and other locations that, again, in terms of this evolution, that obviously this capability provides another way for outlaw cells to expand. Now, the seventh hallmark has just really come into focus in the last decade. Again, heard a great talk today from Peter Commerly on one aspect of that, and that's the notion of deregulating cellular energetics and reprogramming metabolism. Because again, most cells in the body are not proliferating, they're doing their day jobs, whatever those might be, and they have different metabolic needs for energy than rapidly dividing cells. And so we now appreciate that cancer cells turn on alternative energy sources. So you could think of them as hybrid engines where they can tap fuel, varying kinds of fuel to support their cell growth, because both in terms of biomaterials and energy, these rapidly proliferating cells have much different needs than a normal quiescent cell that's part of a society of cells in some tissue. So this, again, provides both cellular building blocks and energy to produce new cancer cells. And this has spawned a whole field of understanding the energetics and metabolism of cancer cells. And again, as we look deeper and deeper, it seems that most tumors have aberrant metabolism and cellular energetics associated with them. And so it seems this is, again, a generic capability. And the eighth hallmark is avoiding destruction by the immune system. And it's long perplexed the field that the immune system is inefficacious at identifying and seeking out tumors. And there have been theories for 50 years about immune surveillance and the notion that the immune system was actually eliminating nascent tumors in some conditions. And it's become clear now that as our knowledge about the immunoregulation has matured and we have better and better tools, that in fact the immune system does, in many cases, seek to eradicate tumors, but that tumors erect a variety of barriers to prevent that. And now there's a whole really ripe field of understanding the diverse barriers that tumors use to keep the adaptive immune system at bay and prevent their eradication. So we think now this happens at different stages in different tumors, but it's that basically there's a lot of evidence now that the immune system does recognize the problem, but it's basically pacified or blocked in varying ways from actually productively killing tumors. And so now a really exciting domain in cancer therapy now is really that we have tools to begin manipulating the immune system to break down those barriers and produce immune attack against tumors. So those then are arguably all are distinctive capabilities, and that we think that many of them, or most of them, are operative in most forms of cancer. And that's why we call them the hallmarks of cancer. So that's then the hypothesis that these 8 hallmarks are sufficient to generate a lethal cancer. Now, the related question is, how are these hallmarks acquired? So how does an aberrant cell acquire all these different capabilities? And we've dubbed these as enabling characteristics, again, which distinguish cancer cells from normal cells and really facilitate this process. And the one that's been evident for 40 years is genome instability and mutation. And it's clear that virtually every cancer cell has aberrations in its genome, which can be range from very subtle mutations and alterations in genes to vast mutational disruption and chromosomal rearrangements. But it's evident now that cancer cells lose a lot of the normal machinery, or they're impaired in the normal machinery that maintains integrity of the human genome, and that then provides a platform for creating genetic diversity. And when some genetic change serves to help the progression of the cancer, it becomes selected again in a Darwinian fashion. So we think that basically the failure of crucial teams of proteins that protect the DNA of the genome from being corrupted, rearranged, and damaged produces or results in the creation of mutations that can convey on cancer cells hallmark capabilities. And so this is an undeniable property of virtually all forms of human cancer. The second one that's come up in the last decade and we now really appreciate is another interesting and important way is that of tumor promoting inflammation. Well, I just mentioned that we think that the adaptive immune system that recognizes antigens and brings in cytotoxic T cells is one half of the immune system, but it was now become apparent is not only is the immune system ineffective in some cases, it's actually worse than that there's now abundant evidence that the immune system can actually help tumors and can facilitate the acquisition of hallmark capabilities. And why is that? Well, the innate immune system is involved in, for example, wound healing. And actually Hal Dvorak, a Harvard pathologist, had made the insightful point decades ago that tumors are wounds that don't heal. And what we now appreciate is that immune cells that normally participate in wound healing inadvertently help cancer cells acquire hallmark capabilities and become more aggressive. Because we now appreciate that these cells can, in the course of wound healing, they induce cell proliferation, they provide survival factors against cell death, they induce angiogenesis, they facilitate invasion as part of wound healing. So a number of these hallmark capabilities are evident in, for example, wound healing as well as in fighting infections. And so these cells, in our view, are misdiagnosed the problem. So they detect a tissue abnormality, they think it's a wound, but it's actually a nascent tumor. And so they infiltrate the tumor and inadvertently help the tumor progress by providing or facilitating acquisition of hallmark capabilities. And that actually leads me into the other realization, is that although the field of cancer research has been focused on the cancer cells, with increasing, we now have the ability to interrogate the entire genome sequence of cancer cells and understand all of the mutations, and much of the field of cancer research has been focused on understanding the drivers that drive these hallmark capabilities and the loss of the suppressors and the brakes that collectively enable these cells to proliferate without control. But it's also now clear that although the cancer cells, with all of their mutations, are important, that tumors are not just bags of cancer cells, but actually they're outlaw organs, and that the cancer cells, as part of their development, attract and corrupt normal cells of the body to facilitate their proliferative expansion. And this has now become, again in the last decade, just functionally very clear about the contributions of these cells, and I've already alluded to them. And so I'll just walk you quickly through that, just to re-emphasize that we think again that there are multiple cells in association with the cancer cells that are contributing to the manifestation of symptomatic disease. Now, of course, the foundation are the cancer cells and the invasive cancer cells that contain the mutations which really render the disease chronic. I mean, one of the problems here, other diseases are degenerative, the cells disappear. Well, these cells persist and expand, and part of that expansion is mediated by that these cells are genetically aberrant. And there's now a new class of cancer cell that's been appreciated that has properties that are more similar to cancer stem cells. And they've been variously called cancer stem cells. They proliferate more slowly, but they appear to be one of the foundations of resistance to disease. They proliferate more slowly. They're just much like tissue stem cells. And this may enable them to really evade some of the targeted therapies, such as the one I described, because they're not proliferating so much, but they have the capability to do so. And so this has become a huge new field of cancer research now, trying to understand cancer stem cells and their interrelationship with the other cancer cells in the microenvironment. So and this is just one example of what we're now appreciating is a phenomenal heterogeneity in tumors, that although there's lots of cancer cells in a tumor, they're not all the same. And this is just the tip of the iceberg in terms of that heterogeneity. But these are at least major classes of cancer cell. But in addition to that, then, there are cells that we think are not mutated. They're simply recruited and corrupted as part of this process. And so the first one is the endothelial cells that form this angiogenic tumor vasculature. As I said, that in most forms of cancer we see an aberrant vasculature in the tumor. And the endothelial cells that form the tubing of the vasculature are present in these tumors, but they're not completely normal. The tumor vasculature is aberrant, but these cells persist and are a common property of tumors. And it's certainly in early stages of tumor development, if you restrict angiogenesis, you severely limit the ability of tumors to grow expansively. And the endothelial cells are supported by periocytes, which are recruited in to help sustain the tumor vasculature and to sustain blood flow. And so both of these cells are functionally important for providing oxygen and nutrients and fuel to the tumor. It's been long appreciated that there are fibroblastic cells associated with many tumors, so-called cancer-associated fibroblasts. And we now appreciate that they're more than just bystanders, but they're actually functionally contributing in various ways to tumor phenotypes. And then, as I mentioned, there are these tumor-promoting inflammatory cells, largely of the innate immune system, although there's also examples from T cells and B cells that are actually promoting and contributing to cancer phenotypes. So that importantly then, these multiple normal cell types are recruited to become components of the tumors, and they help at differing stages of tumor development and progression with a variety of these hallmark capabilities. So the cancer cells don't do it all themselves. In some cases, they recruit and attract directly or indirectly these accessory cells who then inadvertently facilitate the tumor phenotype. And so a variety of studies have suggested that at differing times and differing tumor models, these stromal cells can contribute to, can demonstrably contribute to 7 of the 8 hallmarks. The only one where we have no evidence for stromal support is in the counting mechanism in the telomeres. But other than that, tumor-promoting inflammatory cells, cancer-associated fibroblasts, and angiogenic vascular cells, the endothelials and periocytes, have been documented to contribute to the other 7 hallmarks, but not necessarily in every tumor, not necessarily at every stage of tumor development and progression. But again, the take-home message here is that this is a multi-celled collaboration that's going on. So, but again, the foundation remains the cancer cells, which with the mutations that really render this a chronic disease. And so we've really got these 2 compartments of the recruited accessories to the crime as well as the cancer cells. And so the question then is, how is this happening, and what are these mutations doing? Well, first of all, these mutations in the cancer cells are reprogramming really an elaborate integrated circuit in the cancer cell. And you can make an analogy to the integrated circuits that we all have in our phones and other electronic devices, but which are very, very elaborate. But the signaling circuits within cells are also increasingly well understood, although there's, again, remarkable complexity. But we believe then that cancer involves reprogramming of this integrated signaling circuit in the cancer cell, and that there are different programs within cells that control viability, that control mobility, that control proliferation, that control survival, resistance to cell death. And that changes in these programs conveyed either from paracrine signals coming from the outside or from mutations can reprogram these circuits to convey hallmark capabilities. And so this is sort of an exciting domain now in so-called systems biology, is to try to understand these circuits and how they're rewired in differing cancers. But in addition, there's the broader network of cells in the tumor microenvironment. And so all of these differing cells, the cancer cells, the tumor-promoting inflammatory cells, the endothelial cells and periocytes, and the cancer-associated fibroblasts, as part of their collaboration as an outlaw organ, are communicating with each other. And there's a variety of signals that are transmitted back and forth between these different cell types. And these are just illustrated here are a few of the signals, but there's a vast number of signals that are being transmitted within the tumor microenvironment that contribute to the manifestation of these tumors and of these hallmark capabilities. So, so that's sort of the essence of the concept then, that we have, these acquired capabilities, that there's differing ways to achieve them, and that there's multiple cells that are contributing to these hallmark capabilities, and thus to the manifestation of symptomatic disease in people. So anyway, this was the concept that we laid out, and when we first, I think it's kind of interesting to reflect a little bit on this, is that this was an unanticipated success when we first published this, because we thought it was an interesting idea, but it wasn't really clear that it would be adopted or accepted. But so this article was originally published in the Millennium issue of the journal Cell, which is which is a prominent journal in biomedical science. But I think Bob Weinberg and I had no premonition that it would ever be cited. But actually, it's turned out to be the most highly cited article in the history of this journal. There's over 12,000 references to the original publication, and the sequel to that, where we updated it and refined it a bit in 2011, already has more than 6,500 citations. And so this was, we had no sense of destiny when we published this. We thought it was interesting, it was an interesting collaboration, but it's clearly resonated with the community. And it also, there are several schematic figures in this perspectives that have now become iconic, and so they're widely shown in lectures, they're reprinted, they're adapted for other diseases now, aging and so on and so forth. This whole notion of really concerted capabilities coming together to manifest the whole disease. And I think this is an interesting lesson for students that again, the graphics were, I think, essential for contributing the essence of the idea. And so I'm a great fan of multi-sensory communication, and I think that these figures were crucial to the success in communicating this idea. And it's been downloaded as a PDF over at least 200,000 times, and so by ebook standards, it's been a bestseller. So, given that success, and of course, I guess that's why I'm here telling you about it in this honorific lecture. But so the question then is, it's sort of interesting for the students and others in the audience. Well, how did this free bestseller come to be conceived and written? And so I think it's just kind of fun to look back and think about that, because you might have imagined the two of us were at high tea, or maybe in the pub, or maybe we were in our university offices, and that this is how this idea gelled. But actually, the origins of this were in February 1998, where Bob Weinberg and I happened to be at the same cancer meeting, which was a meeting between the US and Japan cancer societies, where they met in the middle, which is, in this case, was in Maui, Hawaii. And Bob Weinberg and I had never collaborated together. Even though I'd been at MIT, I actually majored in physics and had never met him. And so we didn't know each other. But I'd been to Hawaii several times, and on Maui there's an extinct volcano called Haleakala, which I think is a magical place. And I was standing around in the bar one night with a group of scientists, and I said, well, I'm gonna skip out on lectures tomorrow and drive up to Haleakala, because I think it's just a really special place. If anyone wants to come with me, you're welcome. And everyone said, no, I'm going to the airport, or I've got to give my talk. And Bob Weinberg said, well, I'll go. So basically, although we'd never worked or spent any time together, we skipped out on some lectures and took a long day trip to hike into this dormant volcano, Haleakala. So this is it. So this is, there's a crater, and this is sort of the area around the crater, but it's really a spectacular location. There's a plant called the silver sword that only grows inside the crater. It's a succulent type plant. But yeah, it's really a beautiful and very special place. And so we drove, it took like 3 or 4 hours to drive up there from sea level to 3,600 meters. And then we walked down into the crater to take a look around, and then we drove back down to shore and back to the conference. And since we didn't know each other, we weren't gossiping about the usual stuff that one gossips, and we just ended up starting talking about cancer. And somehow during this discussion, this idea of these hallmarks emerged. And so this was really the inception. Maybe it had something to do with the hypoxia being at 3,600 meters. But in any event, that's what happened. It was during this long day of driving and walking and spending it together. Two of us who'd never spent more than 5 minutes together, we talked about science, and so this concept was born. So basically, we left the meeting thinking, well, this is kind of interesting. Maybe we should keep talking about it. And so, for a year or so, we started occasional communications and brainstorming about this concept. It was a nickname, the Haleakala Review, for a while, and so we began to work on that. And again, as I said, in addition to conceptualizing this in text, I felt that it was essential to conceive the concept in figures, to really crystallize them. And that really related to lessons that I'd had as a student, that I was in part drawn into biology by its inspirational teachers, but also by a textbook that Jim Watson had developed, The Molecular Biology of the Gene, which really had, with every piece of knowledge he wanted to convey, there was a simple schematic. Didn't have to be pretty, didn't have to be artistic, but it did, whether it was the lac operon or whatever, but you had, with every concept that he wanted to get across, there was a simple schematic. And that really resonated with me, and so I felt that we had to have figures that conveyed some of the essence of the ideas. And so I worked on that part in particular, along with developing the ideas in the text. And so in January 2000, this was published, because we had a sort of we were talking about it, and then there was this invitation to write something for this, the January issue of 2000. And so we had then a deadline and put it together and published it. So anyway, that's sort of just an aside on how this idea developed for what it's worth. And and the question then is, well, are there any applications of this to cancer medicine? Are there any applications of this to the war on cancer? And I mean, I think it's arguable that this concept is helping to rationalize the wealth of new mechanistic data forthcoming from the cancer research community. But the question is whether it's applicable to the goal of effectively treating human cancer. And I think the jury for that is still out, but it's one that we've been talking about today during the master class, and one that I'm personally interested in exploring, because it turns out that actually there are targeted therapies that are directed at each of the 8 hallmarks, and actually both of the enabling characteristics of genome instability mutation and tumor promoting inflammation. So of this now wheel of 10 hallmarks and characteristics, we have drugs, we have an armamentarium of drugs that target all of them. And so the question is, will the realization that, because some of these drugs were developed for different reasons, but the realization that they're targeting quasi-independent capabilities, does that help us in any way think about ways to treat human cancer? And of course, so the simplistic war plan, if you were working in the Pentagon, is you would just, of course, have a Hail Mary attack on all of them, and you would just combine everything. And in fact, we were talking today about the history of HIV drugs, where individual drugs you get resistance, much like I presented earlier with that melanoma case, but now with cocktails of HIV inhibitors, there are substantial responses. And so one can imagine that cocktails of hallmark targeting drugs could again add real value. The issue, of course, is that we're facing is one of toxicity, and that this is probably naive because of the toxicity of many of these drugs. So probably, or arguably, what I'm thinking is that we're going to see a future where we're doing sequences of drugs that are targeting differing combinations of therapies. And so you might target a couple capabilities like angiogenesis and invasion and cellular energetics, or you might come in and mobilize the immune system by essentially activating T cells, removing this tumor-promoting inflammation, making the tumors more immunogenic, taking out cell death regimens. And so there's rational ways that you can imagine that you might want to combine some of these hallmark targeting drugs, and that perhaps by, again, sequencing them, layering them, switching from one to another, we'll be able to produce more enduring therapies for human cancer. But at least this is, I think, an interesting hypothesis, and one that, that I'm hopeful will at least have some benefit in some cases. So basically this, again, this project was really a very exceptional, decades-long collaboration, intellectual collaboration with Bob Weinberg at the Whitehead Institute and MIT. And actually there was a now almost extinct species of graphic artists. Everyone does PowerPoints and whatever, but Terry Shoup was a graphic artist who took my crude sketches and PowerPoints and made them into iconic figures. So that's really all I wanted to tell you, except to just leave. And one of the other things I'm hoping to do, and where we're hoping to test some of these hypotheses, is that as I'm involved in a new initiative, which actually drew me to Lausanne, is that we're building a new multi-institutional cancer center based in Lausanne, which brings together a series of institutions in the community which we think have distinctive capabilities. And this includes the University Hospital of the University of Lausanne, where there's a new department of oncology chaired by George Koukos, who's a really terrific physician scientist. We have EPFL, which is my institution, where we have a cancer research institute, bioengineering and chemistry. We're developing a new immunoengineering program, have advanced imaging. And the University of Lausanne has a biomedical research campus with immunobiology and cancer. There's a branch of the Ludwig Institute, which has actually been in Lausanne for many years, but is about to go through essentially a renaissance and refocusing on immunotherapy and immunoengineering. And we've got a powerhouse of bioinformatics, the Swiss Institute for Bioinformatics. And so the nucleus of this multi-institutional venture is going to be a new translational cancer research building, which has been dubbed the Agora Building. It's going to be across the street from the cancer clinics, because we believe that, and I believe from my own experiences, that trying to interact with physician scientists is very difficult, and that proximity is very important. And so the idea is we want to be as close as possible to the cancer clinics. And so we're building this new translational research building that is focused on really trying to bridge the gap between clinical research and basic research. And so what we're doing is we're going to create thematic interdisciplinary programs focused on mechanisms and therapeutic targeting of cancer hallmarks. And that are going to, the idea is to bring together in particular neighborhoods of this building, half floors, clinician scientists, immuno and bioengineers, chemists, biologists, radiobiologists, and bioinformaticians, all of whom are interested in a common problem, be that immunotherapy or metastasis or angiogenesis, so that they will bring complementary expertise and common interests that we believe could help expedite the testing of new therapeutic combinations that could be taken into the clinic. And so that's, and so again, we're going to try to test some of the hypotheses that I raised. And so I'll stop there and thank you.
Thank you very much, Professor Hanahan. I'm always fascinated. This is now the third time I'm listening to an inaugural lecture of the TEFA-Vleerstoel, and I must say that each year it is really a fascinating exploration into an area which for me is, of course, as a non-medical person, I think I saw Bernadette Jansma here, but I don't think we are too many here which are not really in the medical field present here, but it is really fantastic to listen to colleagues from different fields and to explore these areas, which is really done in such a way that it is really fantastic to follow. I find, of course, a number of commonalities, and I would say to researchers, of course, that between Lausanne and, well, I would say Maui in Hawaii, there is a similarity which you see on this picture with these mountains, and I am sure that many of the ideas, as they have developed further in your career, Professor Hanahan, might have to do also with climbing the mountains near Lausanne. We unfortunately don't have that many mountains here. St. Petersburg, maybe, and then, but I don't know whether this will have similar effects here in Maastricht in terms of bringing the same kind of insights with respect to the hallmarks which you described so nicely. Anyway, I think this is a fascinating story, and I must again congratulate Frans Ramakers for having this, our new professor here in this inaugural lecture, and of course congratulate also the Dean and the Vice Dean of the Faculty of Health and Life Sciences. Ladies and gentlemen, may I all invite you now to join Professor Hanahan and to have a drink with him in the Refter here and to congratulate him on this new position. I close this session. Thank you.